Okay, so in this video we're going to look at antibodies and their role in immunity, as well as some of the two types of immune responses, a primary response and a secondary response. And then we'll try to classify some immunity as well. So let's get going. First off, antibodies. There are 5 different classes of antibodies, also termed immunoglobulins, because they're globular proteins. So, and they're denoted by 5 letters. These are the letters of the Greek alphabet: A, G, B, E, and M. Alpha, gamma, delta, epsilon, mu. Those are the Greek letters names. So antibodies are composed of 2 chunks of amino acids. There are 2 long chains, and they are identical to each other. So there's the long chains, and I've got an illustration of this in your notes package. And if you look in the book, page 633, you can see the same thing. So we've got the 2 long chains, they're identical. And then there are 2 short chains associated with them, and they are also identical. And there's connections between these 4 chains. And they're distinguished because here in the heavy chain, there are constant portions. And so this stretch of each heavy chain is constant. So everybody has the same sequence of amino acids in that constant portion of the heavy chain. If it's an A, then it has one set of sequences. If it's an IgG, then it's got a different set of sequences. If it's an IgD, and so on. So we distinguish between them which of these classes of antibodies they belong to, based on the identical or the constant section of the heavy chains. But the reason that antibodies are capable of binding to specific antigens is because that here in what's termed the variable region of both the light chain and the heavy chain, that's where we see lengths of amino acids that can create a binding site that is particular to whatever antigen this antibody is tuned for. So we can bind one antigen here on this variable portion, and a second antigen can be bound on the variable portion of the other half. So that's how they can agglutinate a whole bunch of different antigen-bearing particles, is because the 2 sides of this Y-shaped antibody are capable of, each one is capable of binding to one antigen. And each of the antibody classes has different characteristics and functions. So let's look at those quickly. Immunoglobulin G, or IgG, this is the most abundant kind of antibody found in body fluids. About 80% of all antibodies are IgGs. And they're found in both plasma and in tissue fluids. So we can see them in the blood, we can see them in interstitial tissues, as well as, of course, the lymph. And immunoglobulins G, IgG, is used to defend against bacteria, viruses, and toxins. So if we see lots of these IgGs, then we would expect that that whatever the condition is either bacterial or virus. Well, it could be any of those. So maybe that's not such a helpful thing to know. They also have a unique capacity to be able to cross the placenta. And so because of this, babies are born with antibodies already in their blood and tissue fluids that come directly from mother, are able to cross across the placenta and make it into baby's circulation. That's IgG. Now let's look at IgA. This is the next most abundant, and it's if 80% is the first most abundant, it's going to be hard for anything else to come near that. So 13% of all antibodies are IgA, and these antibodies are found in exocrine gland secretions. Remember, exocrine glands are the glands that have ducts that lead directly to an apical surface, and so breast milk is an exocrine secretion. Tears, nasal fluids, gastric juices, bile, and urine are all places where we would find IgA antibodies. And they are primarily defending against bacteria and viruses. 3 more, IgM, the Muse. They make up about 6% of all antibodies found in body fluids. They are found in blood plasma, so they generally don't leave the blood. And they defend against bacteria, antigens that may be found in food, and this is where we see the red blood cell antibodies, the anti-A and anti-B antibodies. So between IgG, IgA, and IgM, we have a total of, can you do the math? About 99% of all antibodies covered. The rest of them are either IgD or IgE. IgD, they're found on the surface of most B cells, and they play a role in that activation of a B cell. These IgDs do. While IgE is just a small amount of antibodies found in body fluids. They're found in exocrine gland secretions and also in connective tissues on those cells that we talked about back in the tissues chapter 5, as mast cells, the ones that are going to be involved in inflammation. And it turns out that IgE promotes inflammation, and it's IgE that's involved when we see anaphylactic level responses to a toxin or to an antigen. So it can produce super intense inflammatory responses, so intense that sometimes tissue damage occurs, which is what's happening during anaphylaxis. And if you not know what I'm meaning, that's the kind of allergic reaction. This anaphylactic allergic reaction is the one that people have to carry EpiPens so that they can administer to themselves a good-sized dose of epinephrine to help combat what's going on with the IgE. Okay, so what happens then when antibodies are released and they encounter an antigen? We can use red blood cells as an example. Here's a red blood cell. It's got A antigens on it. Here's another one with A antigens. So the first thing that happens is agglutination and precipitation. Agglutination just means sticking together into a large clump, and that's what the antibodies do when they combine, say, this A here and this A here. So now these guys are connected into a clump, and perhaps another set of antibodies comes along here, and it binds these two. So now we've got all three of them hung together in a big old clump. That's agglutination. But if it's a dissolved protein that the antibody is binding to, then it may cause whatever is bearing the antigen, or, and this is maybe more likely with, say, a toxin that's been released, but it can make this dissolved protein, whatever it is, come out of solution so that it is no longer dissolved. And that's referred to as precipitation, so that it falls out of solution, just like rain falling from the clouds. That's precipitation. So agglutination and precipitation are one thing that can happen, or it could cause neutralization. And when we talk about neutralization here, that means that these antibodies will bind to, say, a virus particle or to a toxin that's produced by a bacterium, and it can cover them up so that no part of this this virus or this toxin can make direct contact with anything out there in the body. So it neutralizes the effect of it can't let it be toxic anymore, or it can't let the virus become infective anymore. So that's one set of actions, but when antibodies bind to antigens, there are some other effects as well. And one of the most significant ones is the activation of what's called complement. So when antibodies bind to antigens, then that also makes it possible for the complement proteins that are found dissolved in blood and tissue fluids. It makes it possible for these proteins to also bind. And when the complement proteins bind, then neutrophils and macrophages are both attracted to that antigen. And so the binding of complement really calls in the support. And when we talk about that orientation toward a chemical signature of something, we're talking chemotaxis. Taxis means orienting toward or orienting around a chemical. And so this would be a case of positive chemotaxis because the macrophils and the neutrophils are attracted to the complement proteins that have bound onto the antigen. Complement proteins can also coat the antigen antibody complex, making it easier for phagocytosis to occur, and that's referred to as opsonization. So when let's say a toxin produced by a bacterium gets covered in these complement proteins, it has been opsonized. It's opsonization. Complement proteins can also cause lysis of the antigen-bearing particles. So if it's a bacterial cell, it can cause the cell to break apart. It may cause even more agglutination of any of the particles that are bearing that antigen. It can change the structure, interact with and change the structures of viruses, making the viruses unable to bind to cells and inject their DNA into the cell. And then last of all, it may stimulate inflammation. So it turns out that in most cases, activating complement is the most important thing that an antibody can do toward fighting infection. So it can do that precipitation and agglutination thing. But when we activate complement, we are doing a really good job of finding and binding to the, being able to let the phagocytes come in and just take it out. Okay, so very important component of immunity. So 2 levels of immune response. There's the primary immune response, and there is a picture of this on page 636. It's figure 1621. So in a primary immune response, this is the initial response, the very first exposure to a specific antigen. And it requires the B cells to be activated. So remember that multi-step process. First, we have to get the T cells activated, and then they have to produce a helper T cell that's activated to that antigen. And then the helper T cell has to find a B cell that's also activated toward the same antigen. And then, only after the helper T cell has spewed chemicals all over the B cell, will it divide and become a plasma. So one of the offspring will be a plasma cell. And finally, we get to making antibodies. So if we're looking at the time course in days, so here's 0 days, here's 10 days, here's 20 days, and 30 days. So if we're looking at the time course, it takes a long time to get to the maximum response. Somewhere between 20 and 30 days we hit the maximum, and then we see a tail off. This, by the way, is the number of antibodies. So it's a fairly slow process, and if we think about many diseases as having, say, a 7-day incubation process, So here by day 7, we don't have very many antibodies in, in solution in the plasma. And so the disease can really take off. The disease gets bad, but finally we reach a point, and then the antigen-bearing particles will begin to disappear until they're gone. Okay, so we got sick because the antibodies weren't out there doing their thing soon enough, and a sufficient number of cells became virus infected, or a sufficient number of bacteria grew at the infection site. And so while we didn't prevent the disease, the accumulation of more and more antibodies every hour, every day, that accumulation will fight off the disease. But remember, we produce memory cells. So we produce memory T cells and memory B cells. And many of those memory cells will be able to live for 20 years. Some of them can live for even more. So it just depends. And so the next time then that we encounter one of these antigens, then what happens next is what's called a secondary response. It's a chance for these memory cells to get involved. So let's get this graph back up again. Here's 10, here's day 20, here's day 30. And again, we've got the number of antibodies. And if you recall, we had an increase, there's our increase until we hit peak, and then a decrease. Now, let's look at what happens with a secondary response. With secondary response, do we have to get a helper T cell to be activated? Nope, we don't. Because the memory B cell, when it encounters the antigen, can go immediately to dividing and producing plasma cells that are going to be doing the job of making all these antibodies. And so as they grow and divide, then they're going to be releasing colony-stimulating factors. We're going to see more and more B cells that are sensitized to the particular protein or antigen, and those B cells are going to make plasma cells. The plasma cells are going to begin releasing antibodies very, very rapidly. So that if you look by day 10, we've achieved maximum, almost maximum antibody production. And that antibody production stays high for a very long time. For several months, maybe even. Certainly well past the response, much greater response, and one that is so overwhelming that we prevent the onset of disease. And of course, as these the memory B cells are dividing, they're producing plasma cells, and they're producing more memory B cells that are going to be right there hanging around. So it's a chance to also goose up the level of antibodies. No, I'm sorry, to increase the number of memory cells that are around too. So this is how we can, kind of reinvigorate those memory cells as they are aging, is that we encounter new sources of the antigen, and that stimulates then this immune response that produces a lot more memory cells. So the response here is so rapid, so overwhelming, that here by day 7, we're starting where in our first scenario we start to feel sick. Already we have several times the amount of antibodies that were produced at the same time. And if you'll notice, by the time we reach peak response, somewhere around 10-ish, 12-ish days, somewhere in between 10 and 12 days, peak response, that peak response is overwhelming compared to the response of the in the primary exposure. So this very rapid, very intense response with all these antibodies being produced can, rather than just fight the disease, it will prevent the disease entirely. And then last of all, if we're trying to keep memory B cells around, a type of cell called the follicular dendritic cells, these are cells in the lymph nodes that pick up a bunch of antigens, and then slowly, they will release those antigens, ensuring that there's always a good supply of healthy memory B cells around to fight infection. All right, so let's talk then about classification of immunity. So we can talk about, there are two kinds of immunity. There's active immunity and passive immunity. With active immunity, a pathogen is encountered and we produce a primary response. So we see the production of helper T cells, we see the production of B cells and B cells dividing to produce plasma cells. We notice the new production of antibodies by the body. That's active immunity. And there are two ways that we can get active immunity: naturally acquired, and that just means you get coughed on, sneezed on, you encounter a bacterium, you encounter a toxin or a virus, and in response you develop an immune response and memory B cells are made. With induced active memory, that's what a vaccination is. It's all about providing you a chance to encounter the antigens and then and then produce a whole supply of memory B cells that are going to prevent you from catching whatever it was that you were delivered the vaccine to. And there's several different ways that a vaccine can be, the vaccine can be built. It may be that you actually administer bacteria and virus cells, bacterial cells and virus particles into the body after they've been killed, or at the very least attenuated. If something is attenuated, that means that it has very little chance of causing a bad effect. They've been stunned enough that they can't really infect you, but they're there and present and available for their antigens so that we can produce a population of memory B cells. If it's a toxin that's being used, then it can be converted into some sort of non-toxic form of that toxin. We talk about those non-toxic forms as toxoids. And sometimes we can just isolate the part of the molecule that acts as an antigen and administer what's called a subunit vaccine, because the vaccine only carries these antigens in them. So the goal of vaccination is to create herd immunity, and reduce the transmission of disease. So what do we mean by herd immunity? That means that almost everybody in the population of interacting organisms, almost everyone has earned some kind of active immunity against whatever the material is that's being studied. And so if almost everybody has been vaccinated and has active immunity, then chances of disease being spread from person to person to person are going to be very, very low. So we see, hopefully, a reduction in disease transmission. Okay, so that's active immunity, but there's another way that we can give immunity to someone, and that's called passive immunity, and that's when antibodies are delivered to an individual by some means. So there are two ways, just like with active immunity, two ways you can get passive immunity. The first is naturally acquired passive immunity, and this is when maternal antibodies are transferred to an infant. It can be in breast milk. The first couple of days of breast milk is not really milk at all. but it's a fluid called colostrum that is very, 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 very rich in antibodies, and those are transferred directly to the child, and rather than digested, those tissues are gonna. I'm sorry, I just lost my train of thought. So, that's it. Rather than being digested, the antibodies are going to be moved into the body fluids. They can also jump across the placenta. Remember the IgG that are able to make it from mother's blood to baby's blood. So once antibodies are in the body, they may last for 6 to 12 months in an infant. And so we get a lot of immunity from mama that way. But we can also create induced passive immunity, and that's when we collect antibodies from either human survivors of an infection. So if you think about one of the ways that we treated some of the Ebola, the doctors and healthcare workers who had contracted Ebola, was to give them a solution, inject into them a solution of antibodies that were gathered from one of the, from somebody who's been affected by the Ebola virus. Or we can use antibodies collected from other biological sources, like from horses or from bee cells that are maintained in culture. So we somehow get a hold of antibodies against whatever the antigen is that we're trying to protect the person from, and then deliver those antibodies into that person's blood or lymphatic system. So, for instance, the gamma globulin injections that are given to pregnant women who are Rh negative, but their partners are Rh positive, and there's a chance of having an Rh positive child. And also the injections used to fight a rabies infection, those are also antibodies. Okay, so there you have it. Everything you wanted to know and more about antibodies and how they work. Any questions? Be sure to